Good morning. My name is Nia Lingam, and I'm a summer bioinformatics trainee at the Myon Lab at Mount Sinai. I'm excited to be presenting Biomarker-KG, which is a knowledge graph UI created to facilitate knowledge discovery of data from the Biomarker Partnership. So firstly, what are biomarkers? Biological markers are indicators of biological states and are often molecular or physiological identifiers that can be measured using various techniques and assays from tissues. Biomarkers are crucial to the early diagnosis of disease and tracking disease progression. And the diagram on the right shows the global vision of the biomarker pathway from collection to clinical applications. And recently, biomarkers were in the news as it was found that Alzheimer's disease-specific biomarkers collected from blood in midlife can predict dementia. Specifically, the study found that increased levels of p-tau 181 and decreased levels of amyloid beta 42 to amyloid beta 40 ratio are strongly correlated with dementia, meaning that with a blood test for AD biomarkers, 90% of early dementia cases can be caught. Next, I'll give some background on the CFTE Biomarker Partnership that the Myon Lab is a part of. The project aims to provide a novel systems-level view of biomarkers by aggregating and harmonizing biomarker data from various public resources into one knowledge base. The Biomarker Search Portal is publicly available through GlideGen DCC. Biomarker-KG is an application of the UI that was originally developed for the RiproTalks partnership to provide access to users with no programming skills. And this was a project led by Errol Evangelista, who is a bioinformatician in the lab. RiproTalks-KG is useful for the exploration of associations between birth defects, drugs, and human genes. And similarly, Data Distillery KG is another application of the UI, which can be used to explore the connections between common fund data entities. Users can search for biomarkers using GlideGen's biomarker site, and after a search is submitted, the site generates a table with the biomarkers-related biological entities, and this can include medical conditions, compounds, tissue samples, and variants. Here we see that the searched biomarker in the table is related to prostate cancer, and while the tabular format for viewing related entities is useful, it would be even more helpful to visualize the functional relationships between biomarkers and entities in a knowledge graph format, as seen on the right. Our first step was preparing the data for ingestion into Neo4j, and the biomarker team kindly gave us triplets containing the edges filled with relations for the KG and a list of entity IDs. For the UI, we needed to create nodes from the entities with more information, such as their label, type, and any other metadata. So to do this, I wrote scripts that automated the process for querying the biomarker API, and I retrieved this metadata from the resulting JSON file. And in order to ensure that the data upon which the KG would be made was accurate, we went through some rounds of data cleaning with the biomarker data. And some issues that we found were broad entity labels in the anatomy nodes and multiple biomarkers being linked to multiple IDs, which could be confusing for users, so we decided to collapse these nodes. There was also a gene among the PubChem nodes that we removed, and retrieving the labels for the PubChem nodes required a different approach. And here is a summary of the different assertion types for Biomarker-KG. As you can see, biomarkers have direct relations with all of the other entities, and there are hundreds of thousands of edges and entities within the database. We see that most biomarkers all fall under one of four best classification rules, and some biomarkers are related to multiple medical conditions. After that, we used the ingestion script written by Errol Evangelista to ingest the data into a new Neo4j instance and created a Docker container. As you can see on the right, we can write Cypher queries to create subnetworks of the BKG data within the instance, but these queries can be difficult to write for people without coding experience. We then deployed the UI on the instance and created a schema for Biomarker-KG. And this schema contained a nodes, edges, and UI schema, which would determine how the data would be presented in the site. And after that, the site was ready to be deployed and is now available at this URL. Users are able to search for any of the entities within the database and create subnetworks to explore the relations with the BKG Explorer tool. And there are two ways that users can do this: the one-term search and the two-term search. With the one-term search, users can search for one BKG entity, and the UI will display all the entities that are directly related to it. So for example, on the left, we can search biomarkers by either ID or label, and the site will automatically show the conditions, variants, roles, and tissue samples it's related to. And if need be, we can further fine-tune our results by selecting a specific relation. And on the right, you can see we can search by any other BKG entity type. So in this example, we search for lung carcinoma, and we see all the mutations that have various relations with the condition. And the two-term search is similar, only two BKG entities are searched for at once to see any commonly related entities between the two. So in this example, we searched two example biomarkers, and the resulting subnetwork shows that the biomarkers indicate risks for the conditions in yellow, which are breast adenocarcinoma, lung carcinoma, and ovarian carcinoma. And users can also change the subnetwork display in many ways. We can hover over nodes with the tooltip feature to learn more about them, adjust the graph size with the slider, toggle edge labels, and view the graph in 3 different ways. Users can also filter the subnetwork by target entity type. For example, if we wanted to see only the relationships between the searched biomarker in red and anatomy nodes, we can click on the anatomy icon at the top of the site, and the relation with blood is isolated. Users can also download the nodes and edges from BKG by node type. Nodes and edges are separate to make individual downloads easier, and the site contains some more information for where each node type is from, along with the amount of nodes and edges and the size of the files. BKG also offers a tutorials page which has lessons with screenshots showing how to use the BKG Explorer tool. It demonstrates how to perform the different searches, how to make sense of the results, and the different options for customizing and downloading the subgraphs. And BKG also serves a use cases page with some applications of the BKG Explorer tool. The first use case is for discovering the variants in HRAS that indicate common risk for developing thyroid cancer and skin carcinoma. And using the two-term search, the following subnetwork shows these mutations in yellow. And the significance of these mutations makes sense in the context of the roles in the MAPK pathway. As you can see on the right, HRAS is a GTPase that activates the kinase BRAF, that activates downstream proteins that regulate cell proliferation. Thus, if HRAS were mutated and remained active, the MAPK pathway would remain activated and cell proliferation would be out of control, leading to cancer. And the second use case focuses on breast cancer, as studies have shown that women with breast cancer have an 18% chance of developing a second cancer later in life. So we can use BKG to easily find some other conditions that have common diagnostic biomarkers with breast cancer. The generated subnetwork shows that the BRCA1 mutation diagnoses both breast and ovarian cancer, which has been supported by hundreds of studies, but we also see that the methylation of KLLN can diagnose both breast cancer and renal cell carcinoma. Many studies have indicated that KLLN methylation is a common cause of the two, so this subnetwork is corroborated. And moving forward, we want to add some more metadata to the BKG entities, and we also want to integrate BKG with the glycogen biomarker site. We also want to publish papers about BKG and the original KG UI, along with adding an LLM to help users query the KG and an LLM to summarize the subnetworks to make the site more informative for users. Thank you for your time and for listening to my presentation. I would like to thank everyone in the Mayan Lab for the guidance with this project, particularly my mentors, Errol Evangelista, Ido Diamant, and of course, Dr. Mayan and Mrs. Jenkins for this opportunity. I would also like to thank Dr. Mazumdar, Daniel Masood, and Sean Kim, and the participating DCCs from the Biomarker Partnership for their support as well.